great lead-in with Cindy's talk, and I apologize, I don't have a lot of great photos that are of live animals, because everything I work with usually is dead, but I will try to minimize the gore, because I know we have some young people and people that might be a little sensitive to blood and dead. So just a couple of photos, I'll try to warn you when they're coming up. So I just want a quick quiz. Does anybody know what this creature is? I always like to put up strange animals from the ocean, just to kind of lighten up the thing, get everybody in the mood. Anyone know? Yes? It's a type of squid. What kind? We are talking about squid, right? This is my talk. No. So this is a pig squid. It's a little tiny guy, about the size of a thimble, lives many thousands of feet in the ocean, deep in the ocean. But this is the back end you're looking at, so the little vents that it propels itself with, the two big eyes. So I just thought it was kind of a neat little diversion. So what I am really going to talk about are diseases and health conditions that we see in our porpoises that Cindy probably sees out there, where some of her animals may show up on her shores. So we're going to hopefully start matching up some of these animals to see what they're dying of and get some idea of mortality rate. But for my talk, I'm going to talk really quickly because I have a lot to go over. We're not going to cover every single disease, every single condition, but I just picked a few that I think you might find of interest because they do have some relativity to human health. And one of the things I like to talk about is One Health. So it's animal, human, and environmental health. We're all really connected. So as we go through this, you'll see how it all relates, even to these animals that sometimes we never see. So the talk's going to comprise of just covering some stranding data. Cindy touched on a little bit. We're going to talk about some of these select diseases and health conditions, and then I'm going to talk about antibiotic resistance. That seems to be something that we're hearing about in the news quite a bit, and then I'll wrap it up at the end and give you a summary and how it relates to us. So a little bit of the history of cetacean strandings. They are very commonly occurring. So as you see by the numbers, and they've moved back into the area since the 50s and 60s, we do see them, and I would say pretty much year-round, we get strandings of some sort of porpoises. They've been recorded systematically, marine mammal strandings in this area, pretty much nationwide, but since about the 1980s, it's when it was formalized. Doesn't mean things weren't reported beforehand. Those were more opportunistic, but now we have more of an organized, standardized system by which we record these animals. The numbers that we get, all the data that we accumulate in strandings is a really nice long-term data set because it can mesh with what Cindy's studying in the studies of the life history of these animals, what they're doing, where they're going, what they're eating, and we can record some of the information. We get that from the animals that we see that wash up dead. We also can track spatial temporal trends. Where are these changes occurring? Do we see more animals washing up dead in one area? Do we see changes from year to year? Is it seasonal? Look at patterns. And we can also identify unusual mortality events, and I'll call those UMEs. You hear about those in the news. A little bit of graphics, and I apologize for the dark colors. And I want to acknowledge Jessie Huggins from Cascadia Research loaned me in the next few slides, so I didn't have to pull them all together myself again. But she just gave a talk recently, earlier this year, to a group down south, talking about some of the stranding trends in cetaceans in general. So I'm going to focus a little bit. There is a little bit of overlap with other species, so just bear with me on that. So the most frequently small cetacean that we see are harbor porpoises. In fact, they're the most commonly stranded cetacean of the West, and they account for about 75% of all strandings. That includes as far as cetaceans go. So they are out there. We see a lot of them. They often get called in as, you have a baby orca here. Well, how big is the orca? It's 5 feet long. Well, it's not a baby orca unless it's a fetus. But we get that a lot. So we do appreciate the calls. So we talk through the caller, and cell phones have been great to revolutionize stranding response. We get photos instantaneously. We can say yes or no. But even if it's not an orca, but it's important information. So starting in the so we are seeing some cyclical changes in the graph up here. The orange bars is contrast. The blue is, I think, large whales, but the red bars are all porpoises that we're seeing. So you can see there's some varying patterns. Now, we think the increase in porpoise strandings is due to a variety of factors. It can include true population increases. It could be emerging diseases that are causing more deaths. Funding has increased for stranding response starting in 2000, where we had the Prescott program provide funding so we get a little bit more consistent coverage. Also public awareness. People are learning about these animals, and so they see it, they're more likely to report it. And also there's the ease of reporting, as I mentioned, with the cell phones. Now we're going to focus just on the porpoises, and Cindy did a great job explaining, so I don't have to go through that. Now beginning at about 2000, that's when we really started seeing an increase in numbers of harbor porpoise strandings. At about the same time, or shortly thereafter, in about 2004, we started seeing a really sharp decrease in the number of Dall's porpoises, and I'll show you a map in a minute showing those differences in strandings. So the interesting thing is that stranding patterns for these two species reflect very well what they see on surveys. So if you go out there, you see a lot of stranded harbor porpoises show up. If you go out in the water, you're going to see a lot more harbor porpoises than you will Dalls. So it does, the stranding records can pretty closely mirror what's going on as far as population abundance and location for those species out in the wild. Now the map, and I know these dots are tiny, but I'm just going to tell you in general, the blue dots, starting in the upper left, the blue dots are Dalls, and the orange dots are the harbor porpoise. And the best way to look at this is just kind of go through, we have the 80s in the top left, and then we have the 90s top right, 2000s in the lower left, and then the 2010s in the lower right. And you can just start to see the change. And these are number of stranded animals reported. And then, and I think there are a little bit of sightings in there too. And then the size of the dots represents the intensity. It is a little hard to see here, but you can just see even from back there the difference in color change from the early, for the 80s, where we had more blue dots, fewer orange. And then we get here in the 2010s, we have lots of orange dots, which are lots of harbor porpoises. So we have seen that shift. Now there was a mention about hybrids, and just so happens that we have had stranded hybrids wash ashore. And as it was mentioned, they tend to be the males are the harbor porpoise, and then the female that led to this hybrid was usually the Dalls. And I have a really funny, quick story. When I first moved here years ago, I came from California, where it's all, bottlenose dolphins, and I rarely ever saw porpoise. I had never necropsied a porpoise before, so I got a call from the Olympic Coast National Marine Sanctuary. Hey, can you come out to Port Angeles? It was a nasty January day, and they put me in this little shed over the water, and they said, can you just necropsy this? And we'll come back in a couple hours, and you can tell us what happened. So I said, great. So I established it was a male because that part was protruding. So okay, so I'm opening it up, and I want to do my good job, my first porpoise necropsy, and I'm documenting this, and this is before digital I think it was before digital cameras, so I'm going to age myself a little bit. Taking photos, and I open up the abdominal cavity, and I found this giant tumor. It's huge. Okay, I know what killed it, so I'm writing all these notes down, taking photos, and I'm digging around a little bit more, and all of a sudden, oh my, there's another giant white tumor. I mean, these are taking the entire abdominal cavity. And then I traced them down, and they put it this way, and short story is, they were not tumors, they were reproductive parts. So when these guys are breeding, their testicle size will take up almost their entire abdominal cavity, and then they'll shrink back down when they're not in season, so when they're not breeding. So they can be misleading if you don't know what you're looking at. So on these, the first one that showed up that was recorded was they've been recording the water free swimming before this, but the first stranded hybrid showed up in 2007 in southern Puget Sound. Is the top left photo. And this one had intermediate characteristics between two species. And this was the first non-fetal hybrid that had been found in the Northwest. And then you fast forward to 2011, which is the bottom photo, and that was a female that was found in San Juan Island. She was pregnant, and she also had intermediate characteristics between the two breeds. And this was the first documentation of an actual pregnant hybrid. So the hybrid. So typically when we think of hybrids like a mule, between a donkey and a horse, they are typically not known to reproduce, but this is actually a reproducing hybrid. And there are other species of cetaceans in the world that do reproduce, but it's not well documented if they actually reproduce themselves. So, and I'm sorry for the graphic photos, but these are usually when we see them. Okay, so back to the porpoise stranding, just harbor porpoise in Washington State. So there was a UME, unusual mortality event, that was declared starting in late 2005, early 2006, and it went through 2007, and it included both Washington and Oregon. And there were all age classes involved, and anywhere from calves all the way up to adults. They were widely dispersed throughout the area, so there wasn't really, a clump or a cluster anywhere. And there was no single cause identified, no single cause that could explain all these deaths. And they finally said, okay, well, in looking at all the factors, maybe this is something that's going to be just continuing on. So they closed the UME. There's a panel that decides when it's considered over. But these high mortality rates continued even afterwards. So on this chart, what you're seeing is the blue bars are the outer coast. And I'm going to be focusing more on inner water porpoises, but just to compared to the outer coast, which is blue, and the inner coast, which is red or orange, whatever it's showing up here, you can see that the portion of inland waters or inland strandings tend to be a lot more than outer coast. So here's where the UME was started. You can see that, yes, we've had a little dip down the next few years, but then you see up in 2012, we had a really high spike, and that's here. Right up here, we have really, and interesting with 2012, I'm going to talk about a disease later that we first noticed occurring in these animals in 2012. And right now we're in the midst of doing a big review of all cetacean strandings for North Washington, Oregon, going back from 2003 through 2017. And we've noticed that the patterns of our 6 most commonly stranded cetaceans, including Harbour porpoise, gray whale, and humpbacks, that some of the trends either change or something diverts at the year 2012. Don't know what it is. Still looking at it, could be just all coincidental, but a lot of things seem to be occurring at 2012. The only thing I could find so far is that there was a really, really strong La Nina event during that year. And typically we talk about El Nino, but this was interesting as being a La Nina. So hopefully we'll have more info in the coming years on that. So what can cause Harbour porpoise mortality? And we get all kinds of things that show up in these animals. We can get infectious disease, trauma. These tend to be our 2 most common causes of death. But we have seen some emerging diseases such as protozoa. They're not a bacteria, they're not a virus, they're a different type of organism. Toxoplasma, that's the kitty litter box organism that people tend to shy away from, especially if you're pregnant. Sarcocystis, we have fungal infections, Cryptococcus, I'll talk more about that a little bit later. We've had traumatic injuries such as dystocia or difficulty giving birth, fetal distress, choking, asphyxiation, interspecific, so trauma amongst themselves, also predation and fisheries interaction. And then, so that leads us to the next slide. Fishery interactions and Harbour porpoise are a big concern in this region, particularly in the northwest. There used to be a gillnet fishery off the Nia Bay, and that was a big source of entanglement of Harbour porpoise. We tend to see it a lot inland waters too. So since 1992, when we've been really trying to record fishery interactions very in detail, we've seen about 37 cases, and it doesn't sound like a lot, but when you look at when they occurred, about 86% of those have been just since 2006. And this has been a major cause of mortality, and the problem with it, though, is that we can't always document it. And I had a case that I'll talk about a little bit later. It was a young animal that luckily someone found it and had taken photos of it really close up on the head. Because by the time Central Puget Sound got it, when we necropsied it, the impression marks from the net were no longer visible. So it's like you push down your hand, you have a little dent, you let go, and eventually it's going to fill in again. So that's one of the things that we tend to miss unless you get the animal fresh in photos right away. We may miss those or not be able to record fishery interactions. So it's really been helpful to have people out there, citizens out there with cell phones, we say, okay, quick photo, all over the body and close-ups if you can, so that we can see if there's any indication of fishery interaction. And there are some seasonal trends to this. We tend to see it primarily during the late summer, early fall, when salmon fisheries are active. It's also when we tend to see a lot of our shot sea lions. It's that type of fishery that's going on. And this map is showing some hot spots where we've recorded more cases than other areas. So the intensity red being hot spots, and there's one in the north end of Kitsap Peninsula, and that was a big area. And we've also seen a second one show up way down South Sound, a little bit further down with a second yellow dot at the bottom. And so these areas are ones for us to watch, because as these porpoises are now repopulating these areas, is it something where it's going to be detrimental to them, or are we going to start seeing a lot more deaths due to entanglement? So the three things I'm going to talk about today are going to be fungal diseases. In particular, one, I'm going to focus on cancer that we've seen, and then also talk about antibiotic resistance. So fungal diseases have been around. You know, people get them, domestic animals get them, wildlife get them. Starting in about 1999 to 2000, we started seeing cases of Cryptococcus gattii, typically found in warm tropical areas associated with eucalyptus trees in down in Australia and Indonesia. But it started popping up on the south end of Vancouver Island, which I don't know, last time I checked wasn't really tropical. But we started seeing cases in all kinds of animals, humans, and people and animals that had never left this area, had never traveled outside the Northwest. And it really got a lot of news. Had anyone heard of Cryptococcus? Had heard about the outbreak on the south end? So a few of you had. So we started seeing those in porpoises, quite a number of cases, working with our Canadian counterparts. We'd say, yeah, we got another one, we got another one. And we started necropsying. We'd find these, you know, your typical lymph nodes are about the size of a pea, end of your pinky digit. We would find them, you know, size of volleyballs or basketballs in these animals. So last few years, haven't seen many cases. But what we have started seeing is something called Mucormycosis, which now seems to be called zygomycosis, but I'm going to call it mucor. So it's another type of fungus that is started showing up. So what is this thing? It's a disease. It's caused by the species of an order called Mucorales, and under that order of Mucorales, there are lots of different species of Mucormycosis. And these are cosmopolitan, so they're found all over the place. They're little filament, they're a type of fungi, not quite like a mushroom, but they're filamentous, they're microscopic until they, you know, get into your body. And they are found in decaying organic matter such as leaves, soil, rotting wood, other vegetations, and dung. So the Northwest, the nice forests that we have here are perfect area for these organisms. Well, we had never really seen one. And I'll tell you about when we started seeing it, but I'm getting ahead of myself. So there are about 50 genus of this organism, about 300 species, so there are lots of them. They tend to be rare though. They only occur about 8 to 10% of all fungal infections, at least in humans. And typically you get it from inhaling or getting it directly inoculated into your skin or by eating it. And it can be very aggressive, so if you do get it, it's very aggressive, there's a very high mortality rate. And in most cases I've read about in the human literature, unfortunately the patient has died or has, you know, been very, very sick for a long time. Now in marine mammals it's even more rare, mainly because we probably just don't have as great records on them as we do in humans, but it has been seen primarily in captive animals. In fact, there was a case we just heard about in Harbour seal at the Seattle Aquarium that they've been able to treat successfully in the nose, in a Harbour seal, and that tends to be where they show up in Harbour seals, in the nose. But we have seen a few cases in wild stranded animals until this rash of them that I'll talk about. Now, it's been documented in many, many different species, killer whales, dolphins, you name it, all kinds of seals and sea lions and cetaceans. So it's out there, but it's really not considered a primary pathogen. In other words, it's more of an opportunistic. So if you're not feeling well, your immune system's suppressed for whatever reason, that's where it tends to really take hold. But it's not going to, a healthy individual, it's typically not going to affect you unless there's a really nice portal for it to enter into. So this is more considered an organism, a disease of immunocompromised individuals. For humans, people with diabetes, that have had chronic diseases, that have had tissue transplants, that are on immunosuppressive medications. In marine mammals, we think it's typically those with underlying nutrition or illness, malnutrition, high levels of contaminants like PCBs, DDT. Pregnancy is considered to be immunosuppressing. Traumatic injuries, and interestingly, excess iron, so high iron levels in your body. That's the same thing in humans. But it's, like I said, it tends to rarely occur in healthy individuals, so it's an immunocompromised. So as far as the Northwest, this was first recorded, and we've been doing very, very thorough necropsies on porpoises in this area since about the early 90s, maybe mid-90s, reliably. But we didn't see it until 2012. So as we started seeing these cases, and it didn't really catch our attention until about the year 2015, 2016, and I apologize, these numbers are small. In 2012, we had one. We've had 18 cases so far. I think the numbers add up. Maybe I'm off by one. The column numbers are the most recent. I may have forgotten to update. But the species have been primarily Harbour porpoise. We've had 14 of those, 3 Harbour seal, and our killer whale, which I'll talk about in a minute. You guys probably heard about that one for sure. And these have been, the cause of death has been determined for sure to be this Mucormycosis organism in at least all but 2 cases. And the only reason we couldn't get those is because we couldn't identify it for sure. But we saw it there, we just couldn't get it down to species. The cases have increased over time. So in 2012 through 2014, we had 1 case. They were all Harbour porpoise. And then in 2015, we had 2 show up. 2016, we had 6. 2017, we had 3. And then in 2018, we had 5 that we know of. There could be others. It's just what we were able to document. 2016 was the first year that we had multiple kind of species affected other than just porpoise. We had the killer whale and we had Harbour seal. And the dots on the map show the red are the Harbour porpoise. The blue one that's off of Vancouver Island is where the body of the killer whale was found. And then we have a couple of blue dots up in the Vancouver area, which are Harbour seals that have been affected. Now these are, oh, I'm sorry, I meant to warn you, upcoming is a gross photo, because I want to show you how dramatic it is. On the left-hand side, I mean, these are very, very dramatic, huge swollen masses. So when we open up a Harbour porpoise, and you see, or anything really, marine mammal related that's huge, you're going to think of about 3 different things. You're going to think of fungal, you're going to think of cancer, and you're going to think of a different kind of antibiotic or bacteria called actinomyces. It calls lumpy jaw in cows. And so it's kind of a limited list, and you're always like, ooh, what is this? But in the upper left-hand corner, it shows you one of the ways this mucor can show up as these huge, huge tumors, I mean, big masses. And on the right-hand side, it can show up as little tiny dots throughout there. So I had a professor in vet school who said, never make a microscopic diagnosis without the aid of a microscope. So I always say tentative diagnosis, something really cool, exciting. It's either tumor, cancer, or this actinomyces. Or tuberculosis, I forgot that one, those, that type of bacteria. So anyway, the lesions can present themselves, but they're very, very dramatic. And here's another gross picture coming up. It should be the last of them. So they can affect all the different types of organs. So in the upper right-hand corner, that's a lymph node, and I apologize, there's a short ruler in there, it's about 6 inches long, but that lymph node should be about the size of a pea. So it's very, very dramatic. And what I find most amazing is how long these animals have lived with this nasty stuff in their bodies. I mean, I'm just, they're built to survive. They're built to really go the distance until they just really can't anymore. And some of these animals were pregnant, and that's the most amazing thing. So the next chart is really tiny, but what I'm going to do is just kind of summarize some of the other things that we saw. So I said mucormycosis was determined to be the cause of death of a majority of the animals, but it doesn't mean they didn't have other things going on, because remember I said earlier, usually you're immunocompromised when you get this. So some of the things that we found that they were in poor body conditions, so they didn't eat, they were thin, they weren't doing well. We don't know if it's because of the fungal disease or if it's because they were sick to begin with and then the fungal infection took over. We had some that were pregnant. We had some that were not lactating. But there hasn't been any sign of it transferring to the fetus. Now in the Cryptococcus, the other fungus I talked about, we did actually record it being transmitted from the mom who had it to the fetus. But we haven't seen that happen here. So we don't know, and we haven't seen it recorded in human literature. A lot of parasitism in these guys. Of course, that tends to be the norm for them. And then one porpoise was actually in really, really good body shape. We really couldn't find anything else going on with it. So I might stand corrected when I say it's only immunocompromised. So if you're immunohealthy, we don't think it affects you, but maybe it does. This one porpoise, we couldn't find anything else going on with it, except for the fungus. Oops, these are the ones that... Okay, so what we've been trying to do is use molecular DNA techniques to help identify exactly what strains we have, because you do have cases of it happen in humans. Are they getting it from humans? Are they getting it from the killer whales? Are they giving it to the killer whales? Are humans giving it to them? So this is part of this One Health topic that I mentioned. So we've been trying to send samples off for culture, for DNA testing, and it's not been successful. They are hard to grow. They're very picky about how they grow in the lab, so we don't always get cultures on them. The DNA sometimes doesn't come out very well, so we can't get DNA identification on them. So we're trying different ways, but we have had at least 3 different species so far that show up. Remember, there's 300 of them, so hopefully we don't have 300 different kinds here, but they've identified at least 3 different species by DNA match. And we found one that was something called rhizomucor, but it's the most commonly associated with human mucormycosis. It's also been documented in the Baltic Sea over in Europe. So that tells you that this is a global fungal organism. Now let's talk about L95. How many of you heard about L95? Yeah, okay, so sad story. This animal, as you recall, was tagged off of central Washington coast back in February of 2016. End of March. It was, so it was a 20-year-old young adult male, and at the time they didn't really see anything obvious from what they could see of the animal that it was like, this is something we shouldn't tag. It's really thin. There wasn't really anything to get their attention about that. It was seen a few days later, looking a little bit thinner, and then not seen again until it washed up. Unfortunately, in the rotten condition that it showed up, so that hampers identification. But Dr. Stephen Rafferty from BC did a great job of working up this case and doing the necropsy and the histopathology. And so what they did find is that the tag site had a lot of pathology worse, but one of the things this fungus is known for is it will attack the inside of your vessels. So it'll cause an inflammation, vasculitis, the vasculature system of your body becomes very inflamed, you leak blood. So they did find that in this particular animal. The lungs also had similar organisms that were found at the tag site. So there were fungal organisms at the tag site, fungal organisms in the lungs that looked similar to the tag. And then they decided that the cause of death was due to widespread disseminated mucormycosis introduced at the tag site. Now what we cannot tell is whether the tag introduced it, like the tag had it on it because the tag could have brought it in, or if it was already on the skin and it was inoculated or pushed into the body by the tag. So that can't be determined. So we just know this animal is overcome by this fungal organism. Probably not in the best shape beforehand, knowing how stressed these animals are. So this pathogen found a great opportunity to set up shop, and that's what it did. So what does that mean for southern residents? And back to what Cindy mentioned, these porpoises are great sentinels, canaries in the coal mine, because we can't always, we don't get that many killer whales showing up luckily dead. So the porpoises are a great way for us to get a window view into what's going on in this Puget Sound ecosystem, and especially because it's so urban. So the reason this organism is really of concern, it got our attention finally in 2016 because of this animal dying. But the porpoises have been dying from it since 2012, but again, they don't get the attention. So with the wide distribution of this species in mucor, is that going to mean increasing numbers or increasing risk to the killer whales? Don't know. There are other stressors that the killer whales face, such as insufficient prey, contaminants, noise. So these are all factors that can make these southern residents susceptible or more susceptible to getting affected by this organism. So that's something, and I'll talk about a little study that was done later on when we talk about antibiotic resistance. They did where they sampled the breath of the killer whales and also the surface layer of the water, and they did find mucor there. So, hopefully we don't see more killer whales get affected, but we are continuing to see the Harbour porpoises, so we're going to monitor them. So why are we seeing this now? I mean, that's the $60,000 question. We don't really know. We have some ideas, possibly disturbance of the soil due to urban development. Most of the cases in inland areas are occurring where development has been quite rampant, especially coastal development. And I went back and looked. After the recession in 2008, I looked at kind of just, and this is not scientific, I just kind of looked at when housing development kind of kicked in, and it started back really in earnest, and I mean building new homes, clearing out fields in 2011, 2012. Now I live in Bothell, and it's just far away from there, in the water, farther. But in those areas, I went back and looked when building permits were applied for, and they really started ramping up around 2011, 2012. Now, I'm not saying this is what's causing all this, but it's just another piece of the puzzle that we're putting together. Other causes can be, catastrophic weather events. So in 2015, there was a huge major flooding event throughout Puget Sound, and that's when we saw a number of the cases in 2016. So these are just possibly some causes. Also, could be increased immunosuppression from contaminants. We have more suppressed animals from contaminants or other causes, and also changes in water quality. What factors do temperature changes contribute, or how about salinity, or how about oxygenation? All these things are affecting Puget Sound. We don't know how they affect the fungus and when the fungus is in the water or near the water. So what's going to be next? Well, we're going to continue to try to document these cases very carefully, continue to do our DNA screening to try to find out if other marine mammal species around here are getting affected, what species, to try to look at other tissues. In addition, look at contaminant levels, look at iron levels, because that seems to be a risk factor in humans. There are some inherent conditions that people can get that causes elevated iron levels. Going to evaluate water and environmental conditions. And then looking at, I did contact the pathology labs, veterinary pathology labs in the area. There are two big ones. And to see, hey, did you guys get any cases in dogs or cats, domestic animals, and not yet. So we're keeping tabs on that. And I have a vet lab over in Mukilteo that's been working with us to really help monitor and keep a close eye out for these cases. So that's enough on fungal. Now we're going to come on to another ick organism, which our ick condition is cancer. In general, we don't tend to see a lot of cancer in individual and marine mammals as a group of animals. There are some exceptions. So in St. Lawrence, Canada, an estuary there's a group of belugas that live just in that area. They have a very, 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 very high prevalence of cancer, very high. They also tend to have some of the highest contaminant levels in the world. So that's been an area that we always refer to and try to compare to that, because they live in an urban area in the St. Lawrence waterway. It's very busy, just like it is here. So this particular animal, we had a case show up in 2013, Max Wilton Beach. It was fresh. You can see it's nice and nothing gory about it. Looks pretty good from the side view here, at least. Didn't see anything obvious on the outside. Maybe fishery interaction, nope. But I could tell by her rounded belly, we've got a pregnant animal. This is the time of year that we would see that. We looked at her. We did notice that she was thin. She wasn't our biggest animals that we've seen. We don't see a lot of big, chunky animals coming through because those usually don't strand. But we did notice that she was pregnant. The fetus was estimated about a month long gestation. But man, when we opened her up, she had multiple big firm masses all over the place, particularly in the lungs, and again in her lymph nodes. Because our lymph nodes are our filter system. They're going to collect stuff that's coming through our bloodstream and our lymph system, and they're going to be usually one of the first things they're going to be affected by organisms and by cancer. We did notice some parasites in her abdominal wall, not uncommon. These guys are heavily parasitized just by virtue of what they eat and lifestyle. We worked with our pathologist, Barry Rickman, to look at some possible causes. He did a full histopathology examination of her. He did some special staining, working with folks up in Michigan. They kind of do a lot of staining for cancers for animals. We did molecular type stuff, so looking at DNA, because often we can look at things that are molecular for cancer like we do in humans. Did viral testing, because interestingly, some of the cancers that have been recorded in marine mammals have been, I'm not saying they're caused by viruses, but they've been heavily associated with certain viruses. Morbillivirus, which is the measles virus. We have a big outbreak going on in Washington State right now. And then also the herpes virus, which are associated with some cancers, like Kaposi's sarcoma in humans. So the final diagnosis on this, it was kind of a journey. We first said it was B-cell lymphoma, which is kind of equivalent to some of our Hodgkin's lymphomas in humans that are associated with contaminant levels. Not to say they're necessarily cause, because we can't do experiments to prove it. We have to do a lot of hypothetical associations, but we were able to finally, through all the special staining and DNA work, to get it down that it was something called a multicentric plasmacytoma. It's most closely related to canine or dog plasmacytoma. And went back and looked at this particular cancer, and I'll talk a little bit more about it in a minute, but I wanted to show you some of the contaminant levels. I know this is a boring chart, but the main thing I wanted to show you, we wanted to know the contaminant levels in this pregnant animal with lymphoma. How did she compare to, say, another pregnant animal that maybe didn't have lymphoma? You know, how hard is that going to be to get? Well, it just so happens that about the same time we found this one, got a call from Cascadia, hey, we have a female that's pregnant that looks pretty healthy, about the same pregnancy, about a month along, and she's in pretty good shape, she looks healthy otherwise, nice blubber levels. I said, great, can we get some samples from her? So we compared her, and then we thought, well, how does it compare to a young animal, like a yearling? So we got one that was caught in the fishery, and this is the one where when they first found her, this young animal, the third animal that was in this study, found her, she had impression marks from fishery nets on her face. And by the time we got her to the lab, to necropsy her, they were gone, but we had the photos, so we know that she was a healthy, presumed healthy animal. So the main takeaway from this chart, the black bars are the animal that's the case animal, or I'm sorry, it's the healthy pregnant adult. So that's black bars. The light gray, the lightest gray is the cancer animal. And the kind of medium gray, the tall bars, right here, that is the young animal. So what is odd about this? What do you see? So these are different types of PCB levels. So again, we've got this animal in the middle, the light gray is the affected animal with the cancer. This is her comparison as a same female, same relative age, pregnant, same gestation. What do you notice that's weird? Someone's really keen-eyed here. What do you see that's, you think doesn't make sense?
PCBs are lower in the healthier, younger animal.
Yeah, so the contaminant levels are really high in this young animal. Why do you think that is?
Mom goes from the moms.
Exactly. So same thing in people. So when you're born, if you nurse on your mom, you're going to collect a huge offloading of contaminants from your mom to you. Now, if you're a male, you're kind of out of luck. You don't have a way to get rid of those, really. If you're a female and you go on to breed and nurse a young, then you will offload onto that one. So it was just interesting because, we sort of went, whoa, and then we started thinking, yes, because this was an animal that was weaned, this young animal was a yearling, so it had gotten all its contaminants from its mom. But if you look at comparing the two adults, the pregnant females, in almost, all of the types of PCBs, her levels were much higher, and these were significantly higher, so we did statistical testing on that. So they were indeed higher. Now, it doesn't mean it necessarily causes cancer, but it adds to the evidence. We go to the other type, these are the flame retardants, the PBBEs. Now, interestingly, not every one of these were higher in the affected animal. There's one, let's see, can't see from this. I think there's this one and maybe one other one, I can't tell from this angle, in which the other pregnant female, the healthy one, was higher. But in general, the pregnant female that had the cancer had higher levels of PCBs, and the yearling, again, shot up. The yearling on this one, she didn't have necessarily higher levels than all the two adults. We don't know why. Maybe she just didn't get exposed to it. Maybe her mom didn't have high enough levels of those. But it was a pretty interesting picture. So, talking about the characteristics of this type of cancer, it's pretty uncommon. We don't really see that. It's been described in many animal species and humans, but had not been described in cetaceans before. Now, other types of B-cell lymphomas have, but this particular one had not. So it took a while, and there are other blood-related, these plasmacytomas are blood-related, so they form from the plasma cells, which are involved in our immune system, but there are possible links to environmental pollutants that are noted in humans, and also in other types of cancers have been linked to environmental pollutants in cetaceans, namely those belugas up in St. Lawrence. So in general, with the epidemiology of cancers and cetaceans, we know there's a growing concern. Now, since we put out this report, there are a couple other cancer cases that showed up in porpoises down in Oregon. They haven't been worked up. We don't think they're the same type, but we're not 100% sure yet, so that remains to be seen. We know there's a possible link with environmental pollutants, as I've said over and over. And marine mammals, as you know, are exposed to various types of pollutants. They're very mobile, they move around, they're exposed to urban areas. They also are at the top of the food chain with the fish eating, so they are going to bioaccumulate. They're going to accumulate these contaminants as they get older. And also, they are going to, and they can also, so they'll result in really high levels. So if you're female, hopefully you can offload them to your kids, sorry kids. But if you're a male, there's no way for you to get them. And I think a few years ago in 2000, Dr. Peter Ross from Vancouver reported about the southern resident killer whales, those males that were really highly contaminated. So it's unfortunate. And the last thing I'm going to talk about really quickly is antibiotic resistance. So how many of you have taken antibiotics? How many of you have taken the entire course of them as your doctor said? Okay, I noticed there are a few hands that didn't go up. I won't point you out, somewhere halfway. They weren't admitted. So we hear about this a lot. And people say, well, how bad is it really? Well, I'm not going to say we're a pill-popping nation, but we kind of are. Pill-popping world. We give pills to fix a problem, then we give another pill to fix the problems from that first pill, and so it just kind of adds on. But really, how bad is it? Well, according to some of our big health agencies, it's becoming worrisome. So there was a report that came out about almost 5 years now, that says, yes, it's it's an increasing global concern. And this map is showing you two things. Two ways to show the same problem. So the balls are representing the bigger the circle, sphere, the bigger the problem as far as the resistance cases, deaths due to resistance, antibiotic resistance. And then the darker the country, the continent, the worse, the higher the number of deaths due to resistance. So we are seeing very high rates, particularly in certain settings and certain parts of the world. And they estimate anti-drug resistance, so this covers antibiotics, antifungals, antiprotozoals, not just antibiotics, but it can cause around 700,000 deaths annually. That's a lot. You think about that worldwide. I mean, if you heard Ebola doing that, we'd all be up in arms and going crazy. But this is going to be very insidious. You don't always hear about the deaths that occur. And it may actually become one of the leading causes of deaths for humans by the year 2015. But despite all we know about recording all these stats, that there are huge gaps in surveillance. In other words, knowing how bad it is in certain areas, we don't know. We know in certain countries where we see it a lot of deaths occur, but there are some that are more subtle. So I think it's underreported because it's not always recognized as being related to resistance. And there are lack of standards in trying to, there's no set method to try to record all this data and how to share it and how to coordinate mitigation to it. Well, how does that relate to, aquatic environment? Well, we know that aquatic species and systems can serve as reservoirs for resistance, but it's not really well documented. But I went through the literature and I was quite impressed by the range of organisms that have now been documented as having resistance. Think about going looking at squid or cuttlefish or octopus. I think there's actually an octopus study. They've recorded octopus, they've recorded it in fish, they've recorded it in sharks, of course sea lions, pinnipeds, and marine mammals, the cetaceans. So it's pretty much out there. Now we know that resistance can be created in several ways. So when wastewater discharge that carries antibiotics and the resistant bacteria into the terrestrial waterways. So you water your lawn, you wash your car, it goes down the street into the system that says, oh, don't dump things out here because it's going out to the ocean. It makes its way out to Golden Gardens Park out in Seattle where kids are playing with, little toddlers are playing with their mom in the water that has come down probably a few miles downstream and dumping right into there. And then you wonder why they have closures periodically or E. coli outbreaks. Golden Gardens Park. So I stay clear of that, and I try to tell people, I don't know if I'd swim there, but I get sort of a, what are you talking about? So when it gets into the marine coastline, it can cause disease, And this is really important from a One Health perspective, and reminding that One Health is animal, human, environmental health, because several things can happen when resistant bacteria come into that environment. Well, first of all, you can get sick. So if you've ever been out on a beach closure and you've played around that water outside Golden Norton Park, you can get sick with E. coli. You can also develop further resistance. So the microbes that live in your body can develop further resistance. You can pick up antibiotic or bacteria that are resistant, or you can serve as a reservoir for resistance. And I'm not talking about just humans, but animals too. So you bring your dog down to the beach with your toddler and you go swim in the water that comes in from 5 miles upshore and picked up everything. So it's a disaster waiting to happen. So how does resistance form? Well, we'll talk a little bit about that. Three things that can happen to bacteria when they're exposed to antibiotics. And this is a very simplified version. It's more complicated than this, but these are the top 3. So first off, when you're given antibiotics by your doctor, you hope the bacteria die. That's the purpose of it. You want to kill those bacteria, and that's what most of the time happens. However, sometimes the bacteria, a few will get killed off, and some sit around and they just say, this is good stuff, we're just going to eat it. We're just going to sit around here, we like this antibiotic. We're not going to do anything, we're just going to sit here in your body, we're not going to go anywhere. Or, there are some that are like the tough kid down the street says, you know what, we're going to multiply and we're just going to take over this body or make you sicker. Or we're not going to help you get better. Well, what determines what those bacteria do? Which of those 3 things happen? So there are some things that kind of steer them down to one of those 3 doors. And the 3 most common, again, this is very simplified. One is they can mutate. So these bacteria, they can multiply, and they do it very quickly. So I'm trying to get my little notes here. Okay, so when they multiply, when anything multiplies, the thing that's produced, the offspring, is not going to be the exact same thing as the parents. So you're not the same as your parents. And we see those changes. So we have mutations in us, but most of the time they're harmless. But think about bacteria, they're doing this every few minutes. Okay? So these mutations in the bacteria, such as changes in the structure of their cell wall, can be enough to make it more challenging for antibiotics to kill them. Then, the other thing that can happen is antibiotic concentration. So if the concentration that your doctor prescribes you is not high enough, then those bacteria aren't going to die, or only a portion of them will die. And the third thing is that bacteria can then pass genetic material containing antibiotic resistance onto someone else. So you have a friend that brings you something like, what was this bread, the dough that used to pass among friends? Friendship bread, friendship dough. So someone gives it to you, you change it a little bit, you take some of it off, maybe add some salt in there, and then you pass it on to your neighbor. Well, that bread is not the same, the dough is not the same as you received it and as you've now passed it on. But those little changes are just enough where one bacteria that has resistance can take a little package of genetic resistance genes and pass it on to the next bacteria. So that's how it can be passed on as packages of genetic resistance. So, what do we know about resistance in Puget Sound? There have been a couple of studies that have been published. There's a lot of anecdotal information when we send in animals' tissue samples. Often we get antibiotic resistance studies if you have a culture that's done on a lesion that you see. But as far as formalized studies, there have been 2. One was done by Dr. Joe Gatos at CDOC a few years ago. They looked at rehabilitated harbor seals that were brought into Wolf Hollow. And they looked primarily, though, at pups and young animals that were less than a year. And they looked, I think it was about 46 seals they finally ended up culturing. They had 4 bacteria that they tended to see repeat resistant patterns in. Those are listed up there. So this was a nice study because it looked at harbor seals. It looked at rehabilitation setting. The other study was in southern resident killer whales that Dr. Stephen Rafferty and his colleagues published in 2017. And this is where they took samples of breath in, I believe it was 12 individuals, 12 individuals. And they also sampled the air-water interface to see what kind of organisms those animals could be breathing in every time they take a breath in. And they ended up characterizing the respiratory microbes that they got out of each animal's breath. They identified the pathogens from that blow. They analyzed it for bacteria, fungi, viruses, used genetic methods, DNA methods to identify, and also culture to help identify these organisms. And then they compared the breath samples to the air-water interface samples they did. And they did see multiple drug resistance in some of these organisms from both the surface water and also the breath of these animals. And so it's, kind of thought, what if this is like the fourth, threat to southern killer whales? I mean, diseases aren't really up there with like food shortage and noise and pollution, but if these animals are stressed because of those first three, then this fourth thing, pathogens, can really cause a lot of disease. So how does that relate to what we're doing? So I did a GoFundMe, but it was experiment.com, and it was a crowdfunding challenge. I'll never do it again, but I did learn a lot, and it was quite an interesting experience. And I had always had interest in looking at antibiotic resistance in kind of a multi-species setting in different places around the Sound. So and some of you guys probably are tired of seeing this, saw this and kindly donated, so I want to thank everybody again for that. And we've been posting updates on the experiment.com website. You can look it up and follow the project along. It'll be a year and a half long. We started in October, but essentially what we're going to be doing is looking at describing the presence and extent of antibiotic-resistant bacteria in Puget Sound, the Salish Sea. And we're going to look at differences between two different species, seals and porpoises. So we'll cover two instead of just one species. We're going to look at geographic patterns. We're going to try to collect, we are collecting fresh, dead, stranded harbor seals and porpoises. We're going to do about 70 from each species because we need to get a big enough sample size to analyze this statistically. And what we do is we get necropsies when the animals come in, they're fresh, and we get swabs. And the one thing we're always doing on every single animal, no matter what, is an intestinal swab to look at their gut microbiome to try to see what kind of resistance we see in the intestinal tract, because we are particularly interested in things like Salmonella and E. coli. Now, why did I pick these two species? Well, they're commonly found inside the inland waters. They tend to be more site-specific. They tend to, the harbor seals tend to stay close inside. There are animals on the outer coast, but I try to find species that tend to be more common. They don't tend to wander out of the inland waters as common as other species. They are in greater abundance, and they would be good representation of exposure to human and land-based organisms coming down into the water from land. So the sites that and again, this is not, we're not only sampling from these sites, but these are the general areas that we're trying to get samples from. So I originally had wanted to do like north, mid, and south Puget Sound, but the sample size just wouldn't be enough. So we just divided it in half. So anything from the south end of Whidbey Island north is considered the north part of the study site, and then anything south of Whidbey Island is considered south. And this is a huge collaborative effort. There are other folks involved in this that I didn't have little logos. But it's trying to make use of sample. Animals are going to come ashore anyway. Can we get information that provides us some kind of a quantification of resistance? And what we will be doing is full necropsies on those that we can right away, typically within 48 hours at least being found. If they're starting to smell or they're starting to show some decomposition, we're not using those because we tend to have overgrowth of other bacteria. We want to get them within about, like I said, 48 hours. If they can't be necropsied right away, so they have to be put in the freezer, then we're asking people to take a swab, one of those swabs up in the right corner that we get from the labs, a sterile swab, insert it into the back door there just a little bit, and so we can get a good representation. Put that in this thing. 5 minutes, okay, good, I'm almost done. And then we'll send it off to the lab. And then we're going to come up with susceptibility patterns, looking at the colony, how it looks, and the lab does all that side. And we're going to be doing resistant patterns for each one of those. And then what we want to do is to get information on what percentage or prevalence and what type of antibiotics are there. Are there multiple antibiotics that they're resistant to? We're going to look at key pathogens. We're going to send out the cultures from E. coli in particular. There are human researchers at UW that are very interested in this. They want to know, how does it compare to human strains? Are the humans giving it to the porpoises, killer whales, back and forth? We want to find out which way the traffic is flowing. And the one thing we want to do, it's easy to say yes, there's resistance, or no, there's not, but we want to be able to quantify it to some number to show how severe it is. And this is an easy way to do it. looking at an index. It's very simple. You just, for each organism, you say, look at the number of antibiotics that it was tested for, what proportion of those they showed resistance to. Like, there's typically 16 they screen, including penicillin, amoxicillin, all those. But of those, what percentage? And that'll give us an index number. I think I have like only 2 more slides. So the expected outcome, we expect that we're going to see some multi-species, multi-drug resistance in these animals that we're testing. And we think we're going to see the same type of resistant patterns or similar to the drugs that are used both in human and veterinary medicine. Now we use a lot of the same drugs, in fact, almost identical. There are a few that are used only in animals, but for the most part, veterinary medicine uses human medications. I think we'll see some difference in prevalence proportion between North and South Sound. South Sound doesn't get as much circulation of water. It's, the tide changes are a little bit less, the water doesn't change out as often as it does up north, so we expect to see some differences. We may see some differences in species prevalence of resistance. Not so much in looking at the different age classes. I don't think we'll get the numbers that we want, at least not evenly distributed between different age classes. And we're going to take the strain data that we have. There's an organization, a database that's being formed called Armada at the University of Washington. A couple of professors there that are microbiologists that are starting this. They want to have a data bank for kind of the footprint of each of these, like in humans, what they're doing is every time an organism is cultured, what they do is they genotype it with genetics, and it forms a footprint of that. They put in the database, so the next time someone gets a culture done, they can match it up. It's almost like doing fingerprint matching. So each organism has its own set of resistance. And so we're going to feed that into that so they can compare it to veterinary strains. So just to summarize, we do see an increase in strandings in Harbour porpoise. We are seeing more of them. There are more animals there in the Sound. Mortality rates that prompted the unusual mortality event in 06 and 07 continued. So we think this is going to be the new normal. And so, whether it's just that, it could be that there are more emerging diseases, that there are shifts in the way the population is moving and structured, and Cindy's studies will help us with that, at least up in the north part of the area. There's population increases we've seen. They've come back in after being gone for several decades. We are seeing more interactions with humans, but I think with increased public awareness and reporting, we're getting more cases than we might normally get, so there's a little bit of false elevation from that. So I think at this point is the new normal. We'll see what happens in the next few years. So the last point for us, again, just repeating myself, but the one I really want to do is the last 3 points is that these are really, as Cindy said, these are really sensitive indicators of our ecosystem. And we're unique in that we have a very urban system. We're like Chesapeake Bay, we're like Cook Inlet up in Alaska. So I think it's really incumbent upon us to really be aware of what we're doing, what our habits are. Think about ways that we can be more mindful. We may live in inland, I may live in Bothell, so I'm not on the water, but I'm always thinking about what's going into the water, what's being flushed down the toilet, what am I throwing in the trash. So it's just something to think about, and we'll hopefully have an update in a few years.